Scientists extract and analyze DNA to understand the genetic makeup of different organisms. Considering DNA is contained in infinitesimal cellular structures, the extraction process is surprisingly simple and straightforward. However, as we came to understand, any small mistake could lead to a number of incorrect results. First, we select plant samples and grind them in a microtube using a sterile micropestle. We add a buffer that helps to break down the cells and access the DNA inside, and continue grinding until there are no more visible pieces. We then place the tubes in the centrifuge, a machine that applies a strong force to separate liquids from solid in the tube. The process takes about 10 minutes, so in the meantime, we fill a new tube with isopropanol, a type of alcohol used to further extract the DNA. Once the centrifugation is complete, we remove the tube and can see a distinct pellet at the base of the tube. We remove the liquid above this pellet and place it in the tube with the isopropanol, then invert the solution to mix the two liquids. Next, the tube goes back in the centrifuge, but only for 2 minutes. When it's removed, we discard this liquid because the DNA adhered to the walls of the tube and we no longer need the liquid. We add a solution of 70% ethanol, a type of alcohol, invert it again, and back in the centrifuge it goes. Once the centrifuge is complete, we discard the ethanol because, again, the DNA is on the walls of the tube, then place the open tube in a 60°C block. We leave the tube in this extremely hot machine that allows the liquid to completely evaporate from the tube. We're almost done. After about an hour, we remove the tube and add sterile water, then place the tube on a vortex machine that acts as a rapid massage chair almost to make it easier to extract and read the DNA. Finally, we use nanodrop equipment to analyze the DNA. This machine gives us a graph of the concentration of DNA in the sample. The concentration is depicted in the bottom right of the screen. A concentration of between 100 to 150 with a graph with a straight descending line is considered a good result. That's it, DNA extracted.